everybody says this, but I really mean it when I say how honored I am that you all asked me to speak today. This is up there with one of the biggest honors I've received, especially as a graduate of this program and as someone who is increasingly involved or hoping to be involved in the training of future physician scientists, something I care a lot about, and I'm really excited to talk to you guys today. And the way that I decided to frame things, I'll talk mostly about science, but is to frame it with regard to my path and to tell you a little bit about the science that I was doing along the way and also touch on a lot of the sort of decision points, major obstacles, things of that nature that came up, and hopefully these are things that might be of use to you. And also say up front that I love meeting with and talking to young physician scientists and young scientists. If I can ever be of help to anybody, please reach out to me. So as Sammy said, I was an undergrad at Brown where I did work in neuroscience, and then I was an MD PhD here. I got really specific with the years because I think it's sort of nice for you guys to see when I was doing what. So I started in 2001. Skip and Maggie were here. They were the bosses. It surprised me to find that Skip at that time had only been in charge for 3 years because it just seemed like he knew every detail about everything. It's been a long time for me. I had a meeting a few weeks ago with Skip about something unrelated. I got there a little early. Skip was there a little early. He sat down. He sort of took a deep breath and he goes, "You have a lot of gray hair." And I was like, that's what my 6-year-old says, man. It's not cool. So in my neuroscience PhD here at Penn, I was an NGG student. As Sammy told you, I worked primarily on mammalian synaptogenesis. I was really interested in cell adhesion molecules and how these organize glutamatergic synapses, and they're involved in the initial contact between those. And I was started in January 2004 in my thesis lab. I finished in November to return to the O's. Do the O's still exist? Are they still called the O's? Okay. So I returned to the O's. And one of the first points that I want to make is I didn't know what I wanted to do. I definitely was not someone who was like, I'm going to be a psychiatrist. In fact, I thought that psychiatry was probably not scientific enough for me. I assumed I'd be a neurologist from the time that I had been an undergrad at Brown because neuroscience and neurology start with the same first few letters. So I figured I'd be a neurologist, and neurology is an amazing field. I have close colleagues who are neurologists, but for me on the rotation, sort of like that phenomenon where it's like a rainy day and you visit a college and then you decide like you don't want to go to that college, just the particular mix of things on neurology rotation sort of didn't really jive for me. And my wife, who I met in medical school, she's not a physician scientist, had done psychiatry years before me, and she was saying, "You're going to be a psychiatrist." And I was like, "No, it's not for me." She's like, "They do a little clinical work, and then they go back to their labs. It's going to be great." I said, "I don't think it's for me." But I started looking at it more closely, and I ultimately decided that it was for me. And there were two major things that led me to that decision. One was, this was around 2008, 2009. So this was just a few years after optogenetics had been discovered and was exploding as a field. And so it really seemed to me like this, as a neuroscientist, this was like wide open questions. So much work to be done, so much excitement at that time, and there still is now. And secondly, and this is something I'll come back to a few times, I really made a lot of decisions along the way, keeping in mind that I knew I was passionate about running a basic science lab, asking fundamental biological questions. And so I thought that doing psychiatry was going to be the most likely way for me to reach that path. There were many questions to ask. I was really concerned. I didn't want to burn out. I didn't want to come off that path at all. And so I thought that this was a calculated decision to help me stick with it and to end up running a lab, which was by far the most important thing to me. So I did a psychiatry residency, and I want to first say here that, it's always nice to acknowledge the role of life in your career trajectory. So my wife and I were looking at things at the same time. She was going to become a faculty member somewhere. I was going to match in residency. Penn is really, really good for psychiatry. It's probably not the best place in the country. Maybe now it's getting there, but I don't think it was at the time. And we looked at other places, and she had multiple job offers, and I was looking at multiple residencies, and you have to balance these things. And what I knew was that for my familial orbit, everybody being really excited about what they were doing was essential for all of us to succeed. So that was a big part in me deciding to stay at Penn, and I was very fortunate to have training directors who were really excited for me to be here and were very much willing to sort of negotiate with me with regard to the structure of the residency, and I'll tell you about that in a second. And the next question was about what type of research I really wanted to pursue. So as Sammy mentioned, as a resident, I got involved with this really cool finding at Penn of this new autoimmune encephalitis that happened to attack glutamate receptors, which was my area of expertise, and it had prominent psychiatric manifestations. So I started working on that during residency. So this was a really nice thing about psychiatry. There were a lot of days where I was done at noon. So you can go to the gym, you can go do something else. I would go back and pull charts, and that's what I was doing. And this was a paper in JAMA Neurology, and I was really excited about this work, but I was thinking very hard about what I wanted to do in the future. And I was concerned, seemed to me like the shortest path for me. I thought, okay, like in a year I can be writing a K award, a career development award, quickly start my own lab. I was worried I was going to depend on other people to find these rare autoantibody syndromes in neurology patients. And then I would be the guy like trying to see if they ever happened in psychiatry patients. And I wasn't sure I could kind of do the basic biological work I wanted. And at the same time, I was becoming increasingly interested in sleep, which cuts across all psychiatric disorders. And I also was very attuned to the idea that the person I was going to work with at this next career stage is a more senior person. It needed to be a big lab that could open a lot of doors for me. That was sort of my intuition. And so I was spending a lot of time talking with Amita Sehgal, who right now is at Stockholm, because as you know, the Drosophila circadian work just won the Nobel Prize, and her work was a big part of that. In fact, I think the talks may be happening right now. And I decided to join her lab. She is a world-renowned scientist, world-renowned mentor, which as you guys hear a lot from Skip, matters an awful lot. I will also acknowledge how painful the waiting can be. This is a long path, and I will say I probably loved it more than most in many ways. And there are difficult transition points that I'm sure many of you are experiencing now. So maybe you have a hard time going from med school to PhD, maybe from PhD back to medical school. For me, the hardest point actually was residency to postdoc, because as you can see, they overlapped, and I was a resident during my first year of postdoc. But then they sort of cut me loose in a way in my department. And I took a pay cut and a benefits cut to take what I view as sort of the harder path of having a research career. And this is something that Skip is very attuned to, something I'm attuned to, and something we're trying to make sure doesn't happen to people who are coming up in the ranks, right? At the very least you could do is make sure that you keep a little bit on a forward trajectory in these things that matter in life. But I joined a great lab, and I'm going to tell you a little bit about the science that I started there, which has now launched my own lab. And I was heavily inspired by work that's now a little over 50 years old, okay, that coming out of Stanford, sort of the founding fathers of modern sleep neuroscience in the US, where they first put EEGs on human infants. And they found that human infants not only sleep more, but that their sleep is qualitatively different. And so I was very interested in approaching Amita and saying, look, I'm a developmental neurobiologist. I have a lot of expertise in that area, and I want to apply that to sleep. And they put forward this very influential idea that's been a big part of what I've done, where they said maybe sleep in early life is important for structural maturation of the brain. And this is an adaptation of a really famous figure from that paper where they're showing the sleep amount and by development, and what you see is there's a lot more sleep, but there's a lot more REM sleep in particular. And again, I'll emphasize that life was happening to me as well. So I had kids around this time, and I became even more fascinated by sleep, circadian rhythms, and those sorts of questions in early life. So this is a picture of me in Munich with my son, my second son, Ian, my second kid, Ian. And I asked for a beer bigger than his head, so that was easy to come by. But right off screen here, there's actually a band playing, like really loud brass music, and Ian is sleeping through the whole thing. And these sorts of experiences as a parent, and I think in general, really emphasize that clearly there's something critical going on here early in life, that you sleep and you miss out on these other sorts of environmental interactions. And what controls sleep early in life? What's the neural circuitry? What are the genetics? What's the importance? Can we really prove this hypothesis that had been around for 50 years? Those are the sorts of questions I was really interested in tackling. And indeed, we know that sleep deprivation for things like obstructive sleep apnea can cause outsized and long-lasting neurobehavioral sequelae in humans. We also know that this feature of increased sleep in early life is phylogenetically conserved, telling us that there must be something really important going on here. Young zebrafish sleep more. Young flies sleep more. And David Raisin, an MD PhD neurologist who's now my neighbor in CRB, has shown that there's developmentally timed sleep in C. elegans. Despite there being sleep in all these different systems more early in life, we actually don't know a whole lot about it. So I think that most of you here may appreciate or know that fruit flies are a really exciting model system to study sleep. Just in case you don't, though, in fact, this is work that was done here by Alan Pack and Amita Sehgal, as well as some labs at Wisconsin at the same time. So flies meet all of these behavioral criteria for sleep that are shared across phylogeny. Postural change with behavioral quiescence. It's a rapidly reversible state. It's associated with an increased arousal threshold, so they're harder to wake up. If you sleep deprive them, they rebound the next day, and it's consolidated by the circadian clock. So flies are diurnal. They sleep more at night, just like we do. They have, as I'll show you, a little bit of a daytime siesta. One of the things that I was most fascinated by, though, about Drosophila as a system for sleep is that throughout their lifespan, they largely recapitulate the changes to sleep that we experience throughout our lifespan. So we live about 80 years, flies live about 80 days. A young, newly eclosed fly, one that just emerged from its pupal casing, sleeps more. You're looking at sleep amount in blue. And as they age, their sleep becomes more fragmented. So to me, this makes flies a really compelling system to study sleep and interactions with various pathological states. And today, and what most of my lab works on, is all about sleep in early life. Sammy's work, though, for instance, is looking at sleep fragmentation later on in life and trying to look at models for how we can correct sleep and correct pathological aging conditions. So if you don't know about the fly life cycle, it's one of the really cool things about the fly because it's fast. So from egg to a young adult fly is only about 10 days. And we're working in a tight time window, just the first few days after eclosion, okay? So again, you may wonder, okay, I'll buy that flies sleep. How do you know that flies are sleeping? How do you study this? So in case you're not aware of this sort of thing, we can load individual flies into a glass tube with food on one end. We can pass an infrared beam through the middle, and when they break the beam, we know that they're awake. We know from lots of studies that if they're inactive for about 5 minutes, that correlates to a period of sleep, those other behavioral criteria. You all in back, come on, grab a seat. There's a whole bunch front row, wide open, no problem. So we can load these glass tubes into individual monitors, and then we can load hundreds of these monitors into a temperature and light-controlled incubator. So we can run hundreds, if not thousands, of different genotypes in a single experiment that only lasts a couple days. Right? So there's a nice high-throughput nature to this. When we process the data, I'll show you some traces here, like this, where you see sleep on the y-axis. On the x-axis, you're looking at time of day, day and night. You see this daytime siesta, sleep consolidated at night. And what I'll point out is that this right here, this one graph from a guy, Paul Shaw, is all that was known about sleep ontogeny in fruit flies when I started this project. Okay? Young flies, day 1 after eclosion, sleep more, primarily during the day, and this tapers off to mature adult sleep levels after a few days. In our hands, more like a week, not necessarily 3 days. Okay? So there were so many questions to work on, and the place that we started was, do we know anything about the neural circuitry that controls sleep ontogeny? So we took a candidate-based approach, and we ended up focusing from evidence from that approach on dopaminergic signaling. So dopamine is wake-promoting in flies, just like it's wake-promoting in mammals. And if you imagine some individual tyrosine hydroxylase positive, TH positive neurons firing at a certain rate, then that's pretty low, you're going to get more sleep. If they're firing more, they're going to get more awake. So this is a very simplistic sort of conceptualization. But we wondered, is it possible that this is developmentally regulated? So that a young fly, on the whole, these relevant neurons are less active. That's why the animal is sleeping more, and then they become more active, and that's why they become more wakeful as they age. So the fly brain, in case you don't know, is not homologous to mammalian brains, but the basic neural logic is almost completely conserved in most cases. And we know with a tremendous amount of detail about fly neural circuitry, and we in fact know that there's just a single neuron in either hemisphere that's dopaminergic called PPL1, and this projects to and inhibits a sleep-promoting region in the fly brain that's called the dorsal fan-shaped body, because it's dorsal and it's fan-shaped. That's how we name things in flies, right? And so we took a lot of different approaches to try and understand whether those dopaminergic neurons, those very specific ones, were less active in a young fly, which was our prediction. One approach we took was using a chimeric transcription factor called Kayser, which in response to sustained neural activity translocates to the nucleus and turns on a fluorescent signal. So in that way, we're able to look at fluorescence as a proxy for previous neural activity. And here you're looking at pseudo-colored images of the dorsal fan-shaped body, and what we see is indeed in a young fly, those projections coming from the dopaminergic neurons that input onto the fan-shaped body are less active in comparison to a mature animal. And I'm skipping over most of the data here. This is my postdoctoral work. But it led us to a model that would suggest that you have this basic circuit, and the balance between activity of these groups of neurons sets sleep-wake. But in a young animal, on the whole, these neurons are less active, and so the sleep-promoting region, dorsal fan-shaped body, is more active. And that would be a first example of why an animal would sleep more in early life. So this brings me back, though, to a place that I started at, which is what really is the function of all this. Why would you have this plastic circuit that's programmed to change in activity throughout development? And that's really getting at the heart of the Rothward paper, that seminal paper that I told you about. And now we have this unique handle, right? So in the fly, there are all kinds of neat genetic tricks where we can do things like target just these few neurons I was telling you about, and we can activate just those few neurons in an animal that is moving around, that's awake, that's behaving, okay? So we did that during a very small time window, this critical window of development, and then asked a bunch of days later, which is a long time in the life of a fly, what happens to these courtship behaviors that Sammy sort of mentioned. Now, I'm not necessarily passionate about courtship in and of itself, except that in the fly, we know everything about this circuit in tremendous detail. Every single node of this circuit has been described from sensory input to motor output, okay? So this gave us, we thought, the best opportunity to ask, does disturbing sleep during a critical window affect the development of a circuit that's critical for a complex behavior? And so we did an experiment where the red indicates activating those few neurons during either this critical window early on or in a more mature animal. And what we found was that only in animals that had been sleep deprived via activation of those few neurons during this critical period had a sustained deficit in a single olfactory glomerulus called VA1V that's known to be involved in pheromone-dependent courtship behaviors. Okay? So this seemed really cool because now we can know that if we hyperactivate these neurons and sleep deprive them, there's a sustained behavioral deficit traced all the way down to the single olfactory glomerulus. And then we can say, is it true, in fact, that we're disrupting a normal developmental program? So when we look at the normal developmental trajectories of all of those different glomeruli, which hadn't been done before, to our surprise, it turned out that VA1V in blue, which is outlined here in white, was the one glomerulus that grew far more normally during that exact same time period. So that suggested to us that rapidly growing regions of brain might be most susceptible to sleep deprivation in early life. And one of the things that was completely unexpected for me is the images I was analyzing using this sort of neuropill marker. It's actually a marker of synapse number. So surprisingly now, my PhD work from the MSTP greatest, what's the? Galaxy, right? It turns out that this work I had done was going to dramatically and directly inform the work that I was doing in my postdoc in my own lab, right? Because now it suggests that sleep might be important for synaptogenesis, which is what I had studied earlier. And so it was at this point that I, and actually I'll say about this in a second, sort of unexpectedly began the transition from a postdoc to faculty. So now we're down here. And I want to acknowledge the role of luck and of timing, because that's life. And you all know that if you're in the lab right now. You got to be ready, but luck plays a big role. And I had a paper that came together very quickly, and you get lucky with reviewers because, like, you know, their kid like had a really good piano performance the night before, and they're like a really good mood, it's like really nice to your paper, instead of like the way that it could have played out. And those things are unfortunately reality. And in addition to that, right as this paper was coming out, the School of Medicine was deciding to put a bunch of money behind expanding sleep and circadian neurobiology. And that is essentially how I ended up with my lab at that particular point in time, because our plan as a familial unit was to wait a few years till my wife could make a vertical move, and then I'd be starting my lab. But that's how I ended up staying at Penn, basically. And then I also will acknowledge the importance of being opportunistic. I was constantly on the lookout for opportunities that would be something that could help propel me along. And things like a K Award, you know, one of my reviewers was brutal. He or she hated my proposal. They liked me, hated the proposal. But, you know, there's a lot of back and forth sometimes with NIH, it turns out. And I spent a lot of time with a 35-page rebuttal trying to get my K funded. And as I've talked about with a few people here, Burroughs Wellcome has decided to invest in psychiatry. They have a specific earmark, 2 slots to fund psychiatrists every year right now. And so I was able to take advantage of that, because you can't be just starting your postdoc, and you can't be faculty yet. You have to be right in that sweet spot. How many physician scientists, psychiatrists are there in the country every year at that exact moment? Not that many, probably. It wasn't a huge pool. I think they were only getting 10 to 15 applications a year for those 2 slots. Okay? And then I want to point out this last thing, which I was completely terrified. Okay? When I sort of this was this offer came, and of course I'm going to take it and I'm going to start my lab at Penn. I did not think I was ready. I thought I was going to have like 3 more years to do a postdoc, and I was pretty scared. But what I found from talking to a lot of my colleagues and some of my friends who trained with me here in the MD PhD program at Penn who are now starting their own labs, it seems to not matter exactly how long your postdoc is. You're going to be terrified anyways, so you may as well just jump when the offer comes. Okay? So in my own lab, I'm going to tell you a little bit about that work now. It was this sort of unexpected turn of events where I was interested now in sleep and synapses. And in particular, as a psychiatrist, I was very interested in these neurodevelopmental disorders, which I had been back through all phases of my training. And these neurodevelopmental disorders in psychiatry are increasingly viewed in many ways as synaptopathies. Okay? So these are things that go wrong with synapses during development. And now I'm telling you that we had data to suggest, for instance, that sleep could be a modifier or some sort of risk factor or something like that. So one of the projects that we pursued pretty early on was getting at the sleep-autism link. So sleep and autism are highly comorbid. There's not really any clear mechanistic insight into why so many kids with autism have sleep abnormalities. So we basically decided to take a reverse genetics approach where we said, let's find like every human autism risk gene we can find that has any reasonable homology in the fly. And we decided to do a screen where we would knock down expression of all of these one at a time in the fly brain. And this was work that was actually done by Natalie Gong when she was rotating in the lab, actually during an independent study somehow while she was in, yeah, woo. There you go. Wow, there you go, Natalie. So what she did was, if you guys aren't aware, flies have this really neat set of tools, these binary gene expression systems, where you take a tissue-specific, or in our case, cell-specific GAL4s, in this case, all neurons, and then we can, one by one, express these RNAi constructs and knock them down. So this is a look at the data, where we're looking at sleep amount and all the lines organized by sleep amount. Red and yellow are the 2 controls. So you can see that most things, when you knock it down, sort of fall right near the controls, roughly. But a few were totally off the cliff, right? Dramatic sleep phenotypes and circadian phenotypes. And one of these has become a really compelling story for the lab, where it's a chromatin remodeling gene, not implicated in sleep in any system that we know of. And so we're pursuing that actively. And this is the general approach. And this brings me to a point that I want to highlight, which is my personal opinion is it's a lot easier to go from really basic science and scale up. I think it's a lot more challenging if you are a clinical researcher to sort of scale the other direction. If you have good colleagues and good collaborators, you can do it. And I have 2 different human research projects dealing with insomnia, for instance, that are active right now, and I think it sort of speaks to that. So we looked at human autism risk genes, we found these fly homologs, we knocked them down, and we found some severe sleep phenotypes. Now we're looking at follow-up behavioral studies in flies, but we're also working with other labs to get mice that have mutations in that same gene to look at sleep. And we actually have already now started with a group at CHOP to look for variants in humans. We're really interested in finding kids that may have variants in this gene and other genes and try and see if we can get detailed sleep phenotyping in them. So this is one example of the sort of translational potential, I hope, of the work that we're doing. Okay? So far what I've told you about is the neural circuitry underlying sleep ontogeny, potential roles for juvenile sleep in brain wiring, and then also this reverse genetics approach to identify sleep-relevant autism risk genes. Another area that we've been really interested in is the genetic control of sleep ontogeny, which, as I mentioned at the beginning, it's sort of amazing, right? Like every animal that's known to sleep, which is basically every animal, now including a jellyfish, which don't even have a central nervous system. They have a neural net. This was a recent paper in Current Biology. It really speaks to like what sleep is fundamentally. But we don't know anything about the genetic control of sleep ontogeny, even though basically every animal sleeps more in early life. The work that I've shown you so far talks about the neural circuitry of that. And so, again, we know that humans sleep more, we know that flies sleep more, and we wanted to try and use this genetically tractable system to identify sleep ontogeny genes. So this is another screen that we've done. And I'll say it, the first thing that we did, though, was look at all of the known short sleep mutants in flies, right? So there have been a lot of unbiased genetic screens using fly sleep. It's really cracked open the field of sleep in general. A lot of high-profile papers from Mita's lab and other places as well. Every single one of these animals despite the fact that as a mature adult, they hardly sleep at all. Instead of 900 minutes, they might sleep 200 minutes. When they're young, they still sleep more, okay? All of them do. So it tells you that the genetic regulation of sleep ontogeny, of sleep maturation, is likely distinct from sleep time in and of itself. So this is work that was done by Leela Dilley, who is another MD PhD student who's been in the lab along with Sammy. And we did another screen along with a few other people. And what we did was a tough screen to do, because from a developmental perspective, it was a pain to align everything the way we needed it to be. But we basically did an RNAi screen of neurally expressed genes. And we said, what happens if you're knocking it down? What happens in a young fly and in a mature fly? And because of run-to-run variations in experiments, we wanted to always make sure the young and the mature were being run experimentally at the exact same time. And that was complicated and tedious. So we got only about through 1,000 genes. And you'd hope in a fly sleep screen you might get through 7,000 or 8,000. But this is what the data's looked like, and it spawned a really cool story. So here you're looking at this plot in sort of a unique way. This is young sleep, and this is mature sleep. So as you'd expect, everything clusters to the left, right? Because a young fly sleeps more than a mature fly. We were really interested in things that fall along the identity line or below it, because that would mean that young and old are sleeping the same, right? Or that young is even sleeping more for that, or is less for that matter, okay? And this was the primary screen. We did a secondary library, and then we looked at mutant lines. And Leela's story has converged on a single gene called PDM3. And PDM3 is a PAL domain containing protein. It's involved in nervous system patterning. There are human homologs, and very unexpectedly, there's some SNPs in the human homolog that are already associated with subtypes of autism, which is sort of cool, okay? And this is sort of what Leela's data looks like. So here you're looking at a control for the knockdown and a control for the mutant, all right? Blue is young, black is a mature adult. You're looking at sleep time, time of day. So you can appreciate that blue is above black in both cases, right? A young fly normally sleeps more than a mature fly. When you knock down PDM3 or in a PDM3 het, they're lethal in a null, you completely lose this. This is a really specific and I think super cool phenotype. Basically, these animals never sleep like a baby, right? As soon as they eclose, they're sleeping as if they're a mature animal already. So this is, to our knowledge, the first example of a gene that specifically regulates sleep ontogeny. And so we can also make sure that we're being specific with our findings. So here you're looking at a control, and you're looking at an antibody that's going to recognize in the fly brain PDM3, which is pretty diffusely expressed, by the way. Here you knock down, you behaviorally lose the young sleep phenotype, and protein expression is diminished, and we can re-express PDM3, we can rescue the phenotype, we can rescue expression of the protein. Okay, so we think this is pretty specific here. So harnessing the power of what we can do in this fruit fly system, we next wanted to ask, where is PDM3 in the brain important? It's pretty diffusely expressed, right? So now we're going to use this GAL4 system where you have cell-specific GAL4s. And if you don't know, because most of you don't work in Drosophila, there are libraries of GAL4s that are available. There are intersectional approaches now as well. So there are tens of thousands of GAL4s. And you can get genetic access to manipulate the activity or label or whatever else you want to do to almost any neuron in the fly brain you want, oftentimes with single cell resolution. Okay? So we wanted to see if we could map where PDM3 is involved by just knocking down in specific subsets of neurons. And this was sort of the initial screen that we did, and this particular GAL4 stood out to us because lo and behold, it drives expression in the central complex, which includes this sleep-promoting fan-shaped body I told you about earlier, right? It also includes something called the ellipsoid body. So remember, I told you that the fan-shaped body we think is more active in young flies, less active in mature flies, and that perhaps is why young flies sleep more. Recent work from Mark Wu, who's an MD PhD neurologist who actually trained with Amita about 7 years ahead of me and is now at Johns Hopkins, has shown that the ellipsoid body is also involved in sleep homeostasis. So if you sleep deprive a fly, the ellipsoid body starts firing like crazy. And similarly, in a young fly, it's firing like crazy as well. And so Leela followed this up with a more specific spatial mapping approach where we looked at all of these different drivers that sort of converge in different subsets of neurons within the central complex. And this is a great example of one, R93F07, and there are a few other examples where basically this is the controls, and when you knock it down, you completely lose the young fly sleep phenotype. You may notice that there seems to be a circadian effect here as well. That's something that we're exploring. And this is a beautiful image of a fly brain, and you can see that this is dorsal and then ventral fan-shaped body, and then parts of the ellipsoid body as well. And so we're actively trying to pinpoint specifically where these neurons are relevant. But one of the other things that was so unexpected and exciting about this is work from other labs, to our surprise, had already shown that in PDM3 null animals, so these do not live into adulthood, but if you look at their brains prior to adulthood, this is what they should look like normally. These are actually the dopaminergic innervation to the central complex, right? So I was just telling you about how in young animals, these dopaminergic inputs to the central complex are critical for sleep-wake balance. It turns out in PDM3 mutants, these are completely disorganized, right? So this data suggests a really interesting model where normally you have these dopaminergic neurons, they're firing away this balanced set sleep-wake. In an immature animal, they're less active, sleep center is more active. And we think that when PDM3 is reduced, these neurons are basically hyperactive right away, right? And this is inhibiting the sleep center too much. And that's why this animal, from a sleep perspective, looks like this animal. And this is the idea that we're trying to prove right now that Leela is working on. So in the last few minutes I have, before I'm happy to talk and take questions, I want to talk about one other project in the lab that we've been really excited about. So everything I've told you about so far is talking about the adult fly, right? But as a developmental neurobiologist, I'm really interested in the role of sleep in even earlier developmental periods. And this paper that I've referred to a few times continues to be influential because I had always focused on these few sentences. But it turns out if you look just a few sentences up, they even say a little over 50 years ago, hey, maybe this is also, sleep is also important in utero, all right? But how are you going to study sleep in utero? There's basically no work that's been done on that in sort of premature births. There's some sleep studies that have been done in NICUs, for instance, but very little else. It's just not a experimentally accessible system in mammals, okay? But in flies, despite the fact that thousands of labs probably around the world work on fly sleep, and they've been doing that for a little over 20 years, no one has ever looked in anything but adult flies, okay? You have to be interested in fly sleep, and you have to be interested in development, and that's just a pretty small world. So one of the very first things that I started to do when I began my lab was to see if we could study, basically devise a system to look at whether larvae sleep. And we had a very concrete biological rationale for wanting to pursue this line of work, which is it turns out that flies are unique and the best as far as I'm concerned. So the first wave of neurogenesis is pretty minimal in the embryo. So this is in contrast, for instance, to worms, which are a great model to study sleep. But most neurons in a worm are born in the embryo. You can't study that behaviorally, okay? And in utero, as I've said, most neurogenesis in mammals is occurring very early on, right? How are you going to study that? In flies, it just so happens that 90% of neurons in the adult brain are born during larval stages, right? A widely used behavioral experimental system. So we wanted to ask whether larvae sleep, because if we could show that larvae sleep, we'd have a perfect system to ask questions about sleep and the earliest steps of neural development when neurons are first being born, and we thought that would be really unique. So we created a new system in collaboration with Chris Feng Yen and a very talented graduate student, Matt Churgan, and David Raisin. And we created these things that Sammy coined the phrase larvalage, which is what we're studying here. They were called worm motels by the people who study C. elegans. And all the stuff I'll be showing you was done under infrared conditions, okay? So this is an example of the larvalage, and you're looking at around 20 larvae that we can monitor simultaneously for their behavior. One of the things I want to emphasize is when we were starting this project, we're like, okay, this is going to be great. We're like working with Chris and David, they do this in worms. We're just going to like do it in larvae. We're going to put them in as embryos. And we're going to watch them all the way till they pupate. And it turned out that was incredibly foolish. And just trying to monitor these animals for around 20 hours or 24 hours, which is the entire second instar period, which is when neurogenesis is really getting going, was very challenging. And it really turns out that almost all larval behavioral work is done on the order of minutes. So everything that's published is like 30 seconds, 5 minutes. Longest might be 10-minute assays. And we needed at least 24 hours, okay? So we were able to get that system up and going. Here you're looking at a plot of 6 larvae. This is a heat map of activity. Yellow is active, blue is rest, and you can see that they're tightly aligned. They all molt right around the same time, so they're all staged developmentally, okay? What I hope you can appreciate is there's a lot of transitioning, and it turns out to be pretty stochastic, between periods of rest and periods of activity. Okay? And this is all done by Milan Suparak, who's a research associate in the lab, who's done phenomenal work. So this is an example of, will this play if I do that? No. Of a single video of a larvae, and you're looking at its heat map right here, okay? And you're going to see that it's going to move around a bunch and then have a sustained period of quiescence and then continue moving around some more, all right? All right, so it's moving around. This is, obviously this is time lapse. Okay, well, maybe not obviously. This is time lapse. We take an image every 6 seconds with about 36x speed. And it's completely still for about 2.5 minutes. And then it moves around some more. It becomes completely still again. If you're really astute, you may have noticed that it changed its posture a little bit. So we decided to take this system and ask whether it meets these conserved features of sleep that are shared across phylogeny. This includes, again, a postural change, reversible state, they're harder to wake up, and they should show sleep rebound. So we began with postural change. These are my kids. This is how they sleep every single night, arms interlocked, just like that. Mice sleep with teddy bears, and worms sleep like hockey sticks. So just so you have a little bit of a primer there, okay? And this is a video in real time. It's not the best representation of what we study, but it's a really nice video, and that's why I chose this one. So these are dots of food. This is actually for an assay to make sure that we could tell when an animal's eating, that we weren't confusing things. I'll tell you right now that the postural change I'm showing you and the quiescence are not food associated, okay? But I'm showing you this video anyways. And what you're going to see is the animal is pumping its head. It's eating right now, and it's going to retract its head and enter a period of behavioral quiescence, right? It's a maggot. I mean, it's completely adorable, right? So I'm a psychiatrist, and I'm studying, like, maggots, like eating red food and stuff, but we have good reason to do this, right? We're going to be able to start to ask questions about a biological mystery in our mind, right? And so it turns out that with sustained periods of quiescence, around a half a minute or more, 80% of the time, you see a postural change, okay? So it seems like the longer they sleep, the more likely it is you're going to see a postural change. Is it rapidly reversible, or is this some sort of paralyzed state? Here you're going to see an animal crawling around. It's going to enter quiescence. You're going to see a bright light come on, which is indicated by that, and it moves right away. So rapidly reversible for sure. And then finally, we wanted to ask what happens if we sleep deprive these animals. So we would get a new second instar larvae, let them acclimate. We bother them every 30 seconds. 2 minutes for a 30-second light pulse for around 3 hours, and then we ask what happens. When we do that, you can see that the control animals are sleeping a lot, blue. We're really keeping these other animals awake, yellow. And you can see that we are depriving them of sleep, and in the subsequent 3 hours, they then rebound their sleep, okay? So it seems to meet all of these criteria. We've done a lot of other work that I could tell you about in more detail as well. So the first question, getting more mechanistic to us, was, okay, great, we know all these adult short sleepers. What do they look like when they're larvae, right? These are extremely strong phenotypes. These animals barely sleep. We expected to see a phenotype in larvae, but we didn't, okay? So this is looking at total sleep time in 2 of the most severe adult short sleepers called Sleepless and Fumin. They look exactly like control larvae. Moreover, if you recollect larvae after monitoring them and let them grow up and ask whether their larval sleep is correlated with their adult sleep, the answer is no. It's completely uncorrelated. If it were, it would be organized like this. And adult sleep in flies is sexually dimorphic. So blue males sleep more than females, red. In larvae, that's not the case, right? So it seems like there is at least a partial divergence of the control of sleep early in development compared to later on. And that is, to us, a very intriguing idea. We next wanted to ask, is there at least conservation of some of the neurotransmitter systems? I've told you a lot about dopamine. We also were interested in octopamine, which is the norepinephrine noradrenaline analog, which is also potently wake-promoting in adult flies. And so here, just as I've told you before, we can express various things in cells we want to. This time we're putting in a bacterial sodium channel. You put sodium channels in the membrane only of the neurons we desire, and they're going to depolarize and fire like crazy. And when we did that, it turns out activating dopamine neurons didn't do anything, which was very surprising to us. But activating octopamine neurons potently suppressed sleep, okay? And then we could look at mutants in the octopamine synthesis pathway. If you look at mutants of TBH, they sleep more. That could be because tyramine is accumulating or because you're missing octopamine. So we looked at mutants of TDC, and likewise, they sleep more, okay? And importantly, octopamine neurons and dopamine neurons are there, right? So if dopamine neurons were not present, then it wouldn't be surprising that trying to activate them did nothing. But they're actually intimately involved in a lot of behaviors in larvae. So now we come back to this question, right? Now we have a brand new sleep system. We're the only people doing it. Can we start to look at neurogenesis? So we did the obvious experiment, right? We sleep deprived them. We dissected a couple different time points. Larvae, as it turns out, are a widely used system to study stem cell biology. There are people at Penn who use larvae to do that. And they're much like mammals. So you have a neuroblast, which is the stem cell. It self-renews and gives rise to an intermediate cell, and then terminally differentiates into two neurons. Here you're looking at a larval brain. Green is a neuroblast. Red is a dividing cell. Okay? And what we found was if you sleep deprive these animals, you get about a 15% reduction in the number of dividing neuroblasts during that time. And if you let them rebound sleep, it recovers. So you haven't done permanent damage, right? So this was extremely exciting for us. Of course, we also know that when you're shaking an animal to keep it awake, it's pretty darn stressful, right? So we wanted to try and think of a way to get around that problem. So working with Chris, we created a closed-loop system where we can monitor an individual animal in real time. We can say, hey, are you asleep right now or not? If you're awake, don't do anything. If you're asleep, wake them up. Okay? And then we reverse that system. So now we did the same thing, but if they're active, that's when they get a light pulse. And so we could deliver the exact same amount of light to these animals over the exact same period. So they're getting the exact same stress, but one of them is during sleep, one of them is not. This is a very effective way to sleep deprive the animals that you're trying to sleep deprive. And lo and behold, about a 15% reduction in the number of dividing cells. Okay? So what we think we're able to do here is create this new system where we can now start to dive into mechanistic understanding of how sleep and these very nascent stages of neural development might be linked. And so what I've shown you today and what a lot of the lab is focused on is trying to understand sleep and development using the fly. And I've talked to you about young adult flies, the dopaminergic circuits and synaptogenesis, the reverse genetic screens that you applauded Natalie for, which she deserves, an unbiased screen that helped us identify sleep ontogeny gene, that's Leela's work, and then this identification of a novel sleep platform that we're doing in Drosophila larvae. Okay? And so with that, I want to thank everybody in the lab. I especially want to point out, in addition to the work that everyone's done, Sammy and Leela and Natalie, it's been a huge amount of fun, as I'm sure you can imagine, to have MD, PhD students who are drawn to working in the lab, and also my funding sources. And I want to end by going back to my path and just point out that it's totally worth it. This is the best, all right? I mean, I love my life. I get to spend my time working with brilliant people like all of you. Your research goes in completely unexpected directions. You get to pursue all kinds of interesting things, and you get to be involved and hopefully helping to mentor the next generation of people who are going to pursue things and do it in even bigger and better ways. And again, I'm really honored to have been asked to talk today, and I'm happy to take questions now or talk to you afterwards. So thank you very much.